Hello everyone, I'm Anthony, joined here with Dante, Joey, and Isaiah. And this is our final presentation, explaining everything we did during our lab, mice's antibody response to GFP, inducing and detecting. So, the immune system is the body's advanced defense system. It can find and kill germs, viruses, and abnormal proteins that are trying to get inside. It functions through two interconnected parts: the innate immune system, which provides rapid, nonspecific defense, and the adaptive immune system, which creates a targeted response with B cells and T cells. When an antigen or a foreign protein enters the body, B cells become active and start making antibodies, such as IgG, that react to the antigen. Because of immunological memory, this reaction gets better over time, specifically after being exposed to it more than once.
Like Anthony said, our body has the ability to make antibodies. We can make them against very specific threats. The immune system can recognize specific threats, like the bacteria or viruses, through specialized molecules called antigens. These antigens are essentially labels, and we can put them onto a surface of an invader and use them as unique identifiers. The binding of an antigen to a specific antibody, which recognizes a unique epitope on the antigen, will trigger a specific immune response. This interaction marks the antigen for destruction and will facilitate for its elimination from the body through mechanisms like phagocytosis, complement activation, or neutralization. Antibodies can also deactivate toxins produced by bacteria. Again, memory B cells are long-lived B lymphocytes that will remember past encounters with specific antigens. So, when activated on re-exposure to the same pathogen, they will help initiate a faster and stronger immune response, allowing the body to effectively identify and combat future recurrent infections. Now, how does this relate to our course? Well, for our project, we want to test and see if we can immunize mice against a protein like GFP. We will measure their antibody production response against uninjected, injected, and boosted phases of mice. Again, GFP won't be recognized as a foreign substance, so we need an adjuvant for an immune response. It is a weak immunogen, so we will need the adjuvant to act as the catalyst in the body's immune system recognition as a foreign body being present. That way, the immune system will be triggered and the body will identify the GFP as a potential threat and build antibodies against it. We will be testing this in the future with Isaiah's and Dante's future explanations. Against the uninjected, injected, and boosted blood tests, we will be measuring their antibody production responses on a nitrocellulose membrane.
Hello, everyone. So essentially, I'm going to take you guys here through the methodology, and so our methods are shortened, and this is because our entire project was very long. It was over the course of the entire semester, and because of this, I try to shorten it and essentially highlight what was the most important aspects. So here in our figure one, we showcase our E. coli and our transformation. So what I mean by this is essentially we had E. coli and we were trying to force it to carry a gene of interest, and this was GFP. And so GFP essentially stands for green fluorescent protein, and this was going to be useful through our whole entire project as we were going to try to manipulate a immune response to build antibodies to GFP. And this is going to be done through our animal model, which was mus muscola, which is just mouse, or a mouse. And essentially here, our first step is that transformation, as I was speaking about. And in order to do so, we needed to essentially lyse our cells, isolate our GFP, and then purify it as well. We would then be able to create a vaccination, and this was through our isolated GFP and a adjuvant. Very specifically, we use Freund's adjuvant, and this is lipid polysaccharides, and this is essentially in order to simulate a response, because the GFP itself wouldn't be able to simulate a response or an immune response. So we would then take this injection and we would inject our mouse. Our control was actually collected afterwards, and this was from a related mouse, and this is because we had not previously collected the blood from our mouse that we were using. The mouse that we were specifically using is 1R, and our strain was Black 6 strain. And this was due to the strain being very common and also A being very friendly with us as well. So we use 1FR, so the female right, and the essentially the tag the right is basically just a tag that showcases like which mouse we are using, and this is just a hole on the right side of the mouse's ear. So we would then collect our antibodies, and this would be via a blood collection. So we would either snip off the piece of the tail and collect blood, or essentially create a small hole through the cheek on the vein, and we would collect our blood there. We would then isolate the serum, and this is because, and we would do this by letting our blood sit for 30 minutes and then centrifuge it afterwards and collect the serum, because this is what we needed in order to test the presence of our antibodies. We would so this is all handy dandy, but how would we actually visualize it and see if our presence of antibodies was actually there? This was done via a dot blot, and so the dot blot essentially had a secondary antibody, and this was a conjugate to HRP, and essentially here it's just able to bind to it, and the main idea is that on the left side we have BSA as our negative. Theoretically, we're not supposed to have any BSA antibodies within our mouse. And then on our, so we would do a small dot on the left, and then on the right, we would do GFP. And theoretically, our GFP would bind very strongly. Our GFP antibodies would bind very strongly if it was present. And then, in order to visualize this, we would use chemioluminescence, and we would use specifically a transluminator in order to visually see on our dot plots if our antibodies were present and if they were binding specifically to our nitrocellulose, or on our nitrocellulose membrane, to the secondary antibody.
Okay, so the immunogen we were using is the green fluorescent protein, also known as GFP. It is not native to mice, so it is likely that it would induce an immune response. The protein is initially found in jellyfish, specifically the Equiora victoria. GFP is a naturally occurring fluorescent protein. When exposed to blue or ultraviolet light, emits a green light. So GFP has the capacity to function as a non-invasive marker in living cells and organisms, which highlights its importance in molecular and cellular biology, or in this case, immunology, where we can track the immune response. So how did we administer this to the mouse? So the blood was drawn from the donor mouse and spun in a centrifuge to separate the plasma from the red and white blood cells. The plasma was then inoculated with the GFP protein. We subdued the mice using chloroform to knock it out. After the mice was passed out, we used a purified GFP protein and plasma created using the hydrophobic interactive chromatography and injected into our mice at 2 different dates. So, how did we see if the mouse developed a response? We snipped the tail of the mouse and drew blood from the wound. The blood drawn from the mouse was spun in a centrifuge to separate the plasma from the red and white blood cells. The reason we used plasma was to preserve its clotting factors, which means it was better for immune and protein studies. It's more stable for protein delivery and is faster to process. Due to the weak immune system of GFP, lipid polysaccharides were used as an adjuvant to achieve a stronger immune response. Continuing on this idea, the testing strips were made of nitrocellulose membrane papers. Nitrocellulose binds protein strongly and non-specifically. This allowed for dot plotting directly on the membranes. If the mice made anti-GFP antibodies, these antibodies will bind to the GFP on the membrane. After the introduction of a secondary antibody and substrate, a signal develops showing an immune response. The stronger signal equals the stronger immune response.
So, to give a quick recap on when these developments took place, our transforming of bacteria happened around February 7th to February 13th. And then our first shot and initial blood test was on February 20th. Our second blood test and boost would be on April 3rd. And then our final blood test happened on April 10th, which where we got to have our results observed on April 24th.
So going into our results, our figure 2 showcases our process of transforming the E. coli cells. And essentially here, this just showcases that our E. coli did pick up the plasmid, and it was able to integrate it within itself, and it's essentially expressing our gene of interest. Our figure 3 then showcases that we successfully lysed our cells in order to isolate our GFP product. Our figure 4, this is our specific photo of our successful purification. We were able to get 50 microliters, I believe, of our GFP product. And this was essentially used to divide, or we essentially divided this in order to use it for our first and our second and our booster shot, essentially. So figure 5 shows that we did successfully, do a successful initial inoculation of our adjuvant and our GFP. And this was into our animal model, and this was 25 of each serums, and this would create 50, and this is essentially what we use to inoculate our mus musculus. And then our figure 6 showcases our preparation of our nitrocellulose membrane, as I had spoken about earlier. On the left side is our BSA, which is going to serve as our negative. Our right side is our GFP, which is theoretically we would see some binding to it. And this is, and I had already spoken about on how this is essentially working with our secondary antibody. So our individual results showcase, this is specifically Isaiah's. So on our left side, we have our final bleed. On our middle, we have our initial injection. On our right, we have our non-injected. So it's a bit unclear here because the washes were not able to successfully clean our nitrocellulose membranes. So here on our left, we see BSA, and you could infer that there could be a dot just because of how dirty it is. However, if we look at our GFP, it's very, very, very strong. If we see this left circle here, it's entirely strong, showcasing that there is a presence of our GFP antibodies. Our initial injection shows our BSA again. However, it is a little dirty, so we are not able to essentially say that there was no binding to our BSA just because visually we are not able to see it. But our GFP, again, very, very strong. Our non-injected on our BSA and our GFP seem to not be there at all when comparing to our final and initial injection, which is exactly what we wanted. Again, this showcases that our GFP antibodies were produced successfully. Again, though, the BSA is a bit unclear. So we do have other results as well. So here we see Joey's, and these are clearer as well. So our non-injected on our BSA, no sign of any antibodies binding. On our GFP as well, perfect. This is literally what we want. Our initial showcases that our BSA does not have any signs of binding, and our GFP does. And our final bleed showcases very similarly. However, there is a small little circle with our BSA. This could be due to human error as well, as this is not corresponding with the rest of our results. And we all use the same serum, so theoretically we should all get the same results. This is Anthony's. This is a very, very clear version. Our washes here are done very well. Our BSA is showing up as negative, or is not showing up with any signs of binding, and our GFP does. So our first injection, our non-injected, and our final bleed. This is what we are looking at, and as you can see here, our final bleed is stronger as well. So this is mine. As you can see, this is very clear, and we're able to visually see that our non-injected has nothing on our BSA and our GFP, and our final bleed does have a presence of antibody binding as well with our first injection. Now this is very clear, and we see nothing on our BSA, and this aligns with our theoretical results that we were supposed to get. So, in conclusion, our research group was able to successfully create antibodies using Mus musculus as a animal model, and we were using their immune system. This is able to essentially confirm that with using our dot blot, we are essentially able to confirm that there is no presence of antibody binding within our BSA and are non-injected, which is what we wanted to see in order to see essentially if it was something else that was binding, or if we had dissenting results within each other. And the results, while two of them did seem a bit more unclear, once you got to the other results, it was very, very obvious that we did have a strong production of our GFP antibodies. This was further reinforced by our boost. Our boosted serum seemed to be a lot stronger than our initial injection serum as well, which is theoretically what would happen, because essentially your antibodies on your first injection is going to be a little high, and then after a couple of weeks will die down. However, if you do receive a second boost, your body is essentially going to be saying, hey, this is a threat that we need to keep an eye out for. Let's continue making these antibodies, so your antibodies will skyrocket. So however, both our initial and our boost did show a high affinity for binding. This can suggest high antibody presence. However, this can also be due to us actually having a lot of GFP. We did have 50 GFP in total, and our GFP was bright green. So this can showcase that we did have a lot of GFP. We were not able to measure this with techniques due to specific time constraints. However, this is something that I would put in future directions in order to see if this did play a role in both our initial and our boost having very strong affinity for antibody presence or antibody binding. And yeah, that's pretty much it. I would like to acknowledge Dr. Reyes for helping throughout the project, Vanguard University for this amazing teaching opportunity, the lab staff as well. Another thank you to Dr. Reyes, because without him, we would not have these skills with our animal models. And more importantly, we wouldn't recognize what's truly going on within our immune systems whenever we have a vaccination. So these are literature cited, and that's pretty much it.